Today we are going to study a very important cross, a little bit typical cross, that is known as a trihybrid cross. In monohybrid cross we were studying the inheritance of one pair of allele. In dihybrid cross we were studying the inheritance of two pairs of alleles. But as the name is indicating, in the trihybrid cross we are studying the inheritance of three pairs of alleles. Means here we are studying three characters. Whenever we are studying three characters, means we are studying three pairs of contrasting characters. Okay, see, look here, we are actually going to study here three characters. Say for I have taken three characters out of the 7 pairs of the contrasting characters found in the pea plant. I have taken three characters which will be studied in the trihybrid cross: stem height, seed shape and seed color. But as I have said that when we are studying three characters, that means we are studying three pairs of the contrasting character. How it is? See, what is contrasting character? One is dominant, another is recessive. So if the character is stem height, it is having a pair of contrasting character that is known as tall and dwarf. Tall is dominant, denoted by capital T. Dwarf is recessive, denoted by small t. Say for the another character is seed shape. Then the contrasting pair of trait is round and wrinkled. Round is dominant, denoted by R. Wrinkled is recessive, denoted by small r. Third character, seed color. Seed color, the contrasting pair for this trait is the yellow and the green. Yellow is dominant, denoted by capital Y. Green is recessive and denoted by small y. So here we have considered three characters and we are studying three pairs of the contrasting characters. Okay, now see that in monohybrid cross I have taken one character, means it was actually one pair of the allele was used there, right? And we have crossed capital T, small t, small t. We have taken one character in monohybrid cross. In dihybrid cross we have taken two characters, and the dihybrid cross was actually controlled by two pairs of alleles. Say for capital R, capital Y, capital Y, small r, small r, small y, small y. We have taken this cross, and in F1 generation we have got this one. Here in monohybrid cross, in F1 generation we have got this one. Now trihybrid cross, in trihybrid cross we have taken three characters. You can see here: tall, round and yellow, dwarf, wrinkled and green. Okay, and it is actually controlled by three pairs of alleles. Okay, now you have to view that how we make the trihybrid cross. In the monohybrid cross the checkerboard was made of 4 boxes. In the dihybrid cross the checkerboard was actually formed of 16 boxes, but here it will be formed of 64 boxes. Why 64? Have the answers of this question here, that how a trihybrid cross is made. Here we will be taking, in the parental generations we will be taking three pairs of alleles. Okay, so this is our parental generation. This is our parental generation, means we will be taking the parents here and three characters will be taken: capital T, capital R and capital Y, crossed with small t, small r and small y. Okay, this is tall, round and yellow parent, and this is dwarf, wrinkled and green parent. Okay, now by crossing the parental generation we have to make what? First, the gametes. We have to make the gametes. The gametes will be capital T, capital R, capital Y. Here the gametes form will be small t, small r and small y. Okay, capital T, capital R, capital Y and small t, small r and small y. While crossing these gametes we get which generation? F1 generation. F1 generation means filial generation. This is first filial generation, and first filial generation we get by crossing these gametes. So we will get capital T, capital R, small r, capital Y, small y. Right? You can see here the heterozygous condition, and capital T is dominant over small t, capital R is dominant over small r and capital Y is dominant over small y. So the progeny will be tall, round and yellow. To get to get the F2 generation, what we do the selfing, that is we interbreed the F1 generation. Okay, so capital T, small t, capital R, small r, capital Y, small y. We have done it what? We have done the selfing. This is interbreed by this one, means selfing is done. Now the very important thing is that in the case of the monohybrid cross and the dihybrid cross, the genotype which of the F1 generation was too small, so the gamete formation was very easy. Say for in the case of the monohybrid cross, you have seen that in F1 generation we got capital T, small t, so it was very easy that we have to make 2 gametes by them, like this type. In the case of the dihybrid cross, in F1 generation we were having the genotype capital R, small r, capital Y, small y, and the gamete formation was from this genotype was also easy. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So in the monohybrid cross you have seen that 2 gametes were formed. In the dihybrid cross you have seen that 4 gametes were formed. Okay, 4 gametes were formed. But what about the trihybrid cross? In trihybrid cross, as the genotype is little bit longer, so you will see that 8 gametes will be formed. But formation of the 8 gametes by viewing this genotype is not an easy task. Okay, if you will be trying to make the gametes of this genotype, you will be not able to make it. So I am giving you a method, and by using that method you can easily make the gametes of the following genotype. Say for to calculate the gametes, what you have to do it, that you write like this type, capital T and small t, you like this type, capital T and small t. Then what you do, that cross this capital T and small t with capital R and small r respectively. Okay, then what you have to do, cross this capital R with capital Y and small y, cross this small r with again capital Y and small y, cross this capital R with capital Y and small y, and cross this small r with capital Y and small y. By crossing this you will be getting the 8 gametes. Okay, how will be making it the 8 gametes? 1, 2, 3, 4, 5, 6, 7 and 8. See here, capital T, capital R, capital Y. I am making the gametes here, just concentrate here. Capital T, capital R, small y, capital T, capital R, small y. Okay, then capital T, small r, capital Y, then capital T, small r, small y, capital T, small r, small y. Okay, got it? Then cross this one, small t, capital R, capital Y, like this type, small t, capital R, capital Y, then small t, capital R, small y, then small t, small r, capital Y, and then small t, small r and small y, small t, small r and small y. So I have framed how many gametes? 8 gametes. So this is an easy way to make the 8 gametes. There will be no confusion. Otherwise, if you will be directly forming, may be possible that you can do some sort of error. Okay, so now I have framed 8 gametes from this one. Okay, now exactly the 8 gametes what are formed by this genotype, exactly the same type of the gametes will be formed by this genotype only. So on the horizontal as well as the vertical, in the vertical and the horizontal line, we will be writing 8 gametes like this type and 8 gametes like this type. So 8 into 8 what? 64. So in the F2 generation, the checkerboard which will be formed will be of what? 64 boxes. How it is formed? See here, I am making the format of the F2 generation. Okay, I am making the format of F2 generation like this type. See here, this is F2 generation, and I am making a Punnett square like this type. See, you know very well that here we write the male gametes and here we write the female gametes. Okay, so very first we are writing capital T, R and Y, then capital T, capital R and small y. Third one is capital T, small r and capital Y. Fourth one is capital T, small r and small y. Fifth one is small t, capital R, capital Y. Sixth one is small t, capital R and small y. Then is small t, small r and capital Y. And last one is small t, small r and small y. Okay, I made 8 gametes. And because I have self the F1 generation, so the same type of the gametes can be written here also, capital T, R and Y, T, R, small y, T, small r, capital Y, T, small r, small y. Okay, then small t, capital R, capital Y, then small t, capital R and small y, then small t, small r and capital Y, and then small t, small r and small y, right? I have made a format here, and you will be able to see that 64 boxes will be formed here. Okay, see here. I will be not filling the entire checkerboard because it's little bit time consuming. I will be liking to fill one row and then you can try yourself, okay, so that the time will be less consumed. See here, I am crossing this with this, this with this, and so on. Capital T, capital T, capital R, capital R, capital Y, capital Y. This one, capital T, capital T, capital R, capital R, capital Y, small y. Then this one. Then this with this. Then this one with this one. Okay, then that with that one. Capital T, small t, capital R, small r, capital Y, and this with this one. Capital T, small t, capital R, small r, capital Y, small y. I made a format. You can see that 1, 2, 3, 4, 5, 6, 7 and 8. 8 in one row. So there are total how many rows? 1, 2, 3, 4, 5, 6, 7 and 8. So 8 into 8 means 64 boxes will be formed. Okay, so this is the format. I have not filled here the entire one, right, because it will take at least 15 to 20 minutes. So you can try at your home. I have made the format. You can just try it like this type, this with this and so on. Okay, now the question arises, like the monohybrid cross and the dihybrid cross, can we calculate the phenotypic ratio and the genotypic ratio of a trihybrid cross? So definitely it's genetics and we can do it, but many students leave the phenotypic ratio and the genotypic ratio of the trihybrid cross because it's little bit lengthy, but at least because you are viewing this video, you must know this thing. Okay, so we are going to calculate the phenotypic ratio and the genotypic ratio of the trihybrid cross, but by the traditional method, by making this box of 64 boxes and by viewing each and every box and then calculating the phenotypic and the genotypic ratio is little bit a tedious one, right? So we will be not calculating the phenotypic ratio and the genotypic ratio what was calculated earlier by the orthodox methods. We will be using some mathematical formula to calculate the phenotypic and the genotypic ratio. So have a look. To save our time, we are using some mathematical method to calculate the phenotypic ratio and the genotypic ratio of trihybrid cross. See here, very first we are going to study the phenotypic ratio. Okay, so to calculate the phenotypic ratio of a trihybrid cross, we use the formula 3:1 to the power of n. But as we know that in monohybrid cross we have used the power 1 here, in the dihybrid cross we have used the power 2 here, but in trihybrid cross we will be using the power 3 here. Okay, and then we will be calculating the phenotypic ratio. So how we will be calculating? 3:1 into 3:1 into 3:1. Now to make the things easier, what we have to do, first multiply these two and then whatever we get, multiply that with this one. Okay, so calculate this one. This is multiplied by these two and this is multiplied by these two. Okay, so let's have a look. 3 3 ja 9 and 3 1 ja 3. Okay, and 1 3 ja 3, 1 1 ja 1. Okay, so we have got the ratio 9:3:3:1. Now we have to multiply with this, that is 3:1. That is, we have to multiply 3 with all these 4 digits and 1 with all these 4 digits. The ratio what we'll be getting, that will be the phenotypic ratio of a trihybrid cross. Look here, 9 3 ja 27. Okay, 3 3 ja 9, 3 3 ja 9 and 3 1 ja 3. Okay, then multiply this with all these 4 digits. 1 9 ja 9, 1 3 ja 3, 1 3 ja 3, 1 1 ja 1. Okay, we can also rearrange this ratio. Like this type. 27:9:9:9:3:3:3:3 3:1, right? To remember this thing, how we will be learning this phenotypic ratio of a trihybrid cross? 27, 9, 9, 9, 3, 3, 3, 1. Learn like this type, okay? 27, 9, 9, 9, 3, 3, 3, 1. So this is what? This is the phenotypic ratio of a trihybrid cross. This is a phenotypic ratio of a trihybrid cross, okay? So this is little bit easier method. Instead of viewing the 64 boxes, writing the names of the progenies and then writing the ratio, it may cause some sort of error. To avoid this thing, we have used the mathematical method instead of the traditional orthodox method, okay? Now we will be going to calculate the genotypic ratio of trihybrid cross. It is very, very typical one. It's very typical. See here. Now again, because by calculating it by the traditional method, it is little bit a tedious and a cumbersome process. So what we will be doing to calculate the trihybrid cross genotypic ratio, we will be using a formula. We will be using a formula 1:2:1 and power is n. But because it is a trihybrid cross, so what we will be doing? We will be using the power 1:2:1 to the power 3, because we have taken how many characters here? 3 characters. What means? That is 1:2:1 into 1:2:1 into 1:2:1. To make the thing easier, what I have did, that first I will be taking out, I will be multiplying this 2, 1:2:1 multiplied by 1:2:1, and whatever I will be getting, then I will be multiplying that thing with this one, okay? So see here, this 1 is multiplied by these 3, this 2 is multiplied by these 3, and 1 is multiplied by these 3, okay? So have a look here. 1 1 ja 1, 1 2 ja 2, 1 1 ja 1, okay? Then multiply this with these 3 digits. 2 1 ja 2, 2 2 ja 4, 2 1 ja 1, okay? Then multiply this with this. 1 1 ja 1, 1 2 ja 2, 1 1 ja 1. I have got this one by multiplying these 2. Now cross with this one. 1:2:1. Now what you have to do is that you have to multiply this digit with all these digits. You have to multiply this digit with all these digits, and you have to multiply this digit with all these digits. The thing what you will be getting, that will be the trihybrid genotypic ratio. So have a look here and just concentrate it. Now see here, if you multiply 1 by all these value, you will be getting the same value. So I am writing here 1:2, okay? I am writing 1:2:2:1:1:1:2:1, okay? You have got it. I have multiplied this with this all digits. 1 1 ja 1, 1 2 ja 2, 1 1 ja 1, and so on, okay? Now see here, from here, now multiply this 2 with all these digits, okay? So 2 1 ja 2, 2 2 ja 4, then 2 1 ja 2, 2 2 ja 4, 2 4 ja 8, 2 1 ja 2, 2 1 ja 2, 2 2 ja 4, 2 1 ja 2, okay? See here, got it? Now what I have did, I have crossed this with this one, okay? Now cross multiply this with this one, so I will be getting the same one, okay? 1:1:1:1:1, okay? So this is your trihybrid genotypic ratio, right? 1:2:1:1:2:1:2:4:1:2:4:8:2:2:4:2 is to 1 is to 2 is to 1 is to 2 is to 4 is to 1 is to 1 is to 2 is to 1. Don't try to mug up this thing, okay? What you have did it here, 1 is to 2 is to 1 was crossed with 1 is to 2 is to 1 was crossed with 1 is to 2 is to 1. Okay, so first what I have did, I have multiplied these two and I have got this value. Then I have multiplied this value with this one. Then I have multiplied this with this value, this with this value, this with this value. I have got this one. So this is what trihybrid genotypic ratio. Okay, so if 1 or 2 times you will be doing practice, you will be able to write the phenotypic ratio and the genotypic ratio of trihybrid cross also. Okay, so I will be coming with new videos in the coming time which will be based on genetics and other topics. And also students, if you are facing any problem regarding botany, zoology or any topic of the biology, you can just ask me in the comment section. I will be trying my level best that whenever I will be becoming free, I will be trying to give answers to your questions. Share my video, like my video and also subscribe my channel. Thanks a lot.